we're concentrating on topic 1, which is really revision of living systems, which is the introductory chemistry and biology subject. And the notes are extremely comprehensive in that regard. So we have the audio from the lovely Anne Manning, and we also have some annotated notes. So I think they're very good. So what we're going to do is, in these tutorials, we're going to go through some problems for each one. And I'll give you some homework, inverted quotation marks, not overly laborious homework to do before the next tutorial, which we'll have fortnightly. Let me see. Michael, you're based in Melbourne, aren't you, Michael? Yeah. Yep. So you're not working at Vintage this year. Oh, nice. Which part? Yeah, great. Oh, brilliant. What winery are you moving to? What Bass Phillip? Hitting the Bass Phillip Express? Oh yeah, cool. Yeah, nice. They do a big range of wines. That'll be very interesting. Well done. All right. So, and Alex, you're in Melbourne, aren't you? Or are you commuting down from Rutherglen? What are you doing? Is that a silly question? I feel that might be a silly question. Yeah, in Melbourne. Very good. So we have grapes coming into the winery as of tomorrow. So the assessments which we're going to go through now partially require the tracking of the wine. So if you're not working a vintage at the moment, or you aren't this vintage, or this year, you can track wines in the winery here. Just make time with me and we'll work out the logistics, and that goes for any student in the subject. Tomorrow afternoon, we're running a series of pracs. I know, Alex, you've done those already. But Michael, I think you'll still need to do some introductory practicals, wine chemistry practicals. If you're available tomorrow afternoon, that'd be great. Otherwise, we can try and fit them in another time, but there's also a self-guided one, which I'll be putting up as well, which is very easy to follow, but it's always good to come in and do it as well. Bit of fun. We concentrate on sulfur, pH, TA, alcohol by a bulliometer. Yeah, we had, and also an enzymatic analysis of sugars. Yeah, they are, Alex. Yeah. Oh yeah, you got to do the pracs, don't you? That's right. Yeah. So running some pracs tomorrow afternoon, as well as doing a crush. So it'll be a very real life analysis session, or we can do it further down the vintage. But if you can come in tomorrow afternoon from 12:30, that'd be great. Yeah, cool. Yeah, no worries. Well, I'm going to do some with Andrea Walker as well, potentially. So you can come in on those. But you'll find these opening ones fairly straightforward, I imagine. They're still worth doing though, because we do it. This workshop is assessed, but pretty much by rocking up and going through them, you'll get 100%, and they're all based around just processes of standard analyses, doing it the right way, working out how accurate we have to be for some things and how inaccurate we can get away with for others. So assessments. So Michael, we'll see you tomorrow, hopefully. So Let's have a look at the assessments here. Let me click on subject outline. So we have the exam, which is sort of standard 2-hour exam, and we're going to be doing heaps and heaps and heaps of revision related to that. Where did that go? There we go. So the assessment, the exam is quite challenging, but you'll be well and truly prepared before we do it. Plus previous year's exams, so you can work out what to do. So we've got a 2-hour written exam, 120 marks. We have a practical logging exercise. Have a look. Yeah, so we've got the exam, that's assessment 2. We have the practical exercises, which is assessment 1, and they're going to be based on the preliminary pracs before the tutorial, before vintage, the traditional vintage period, and a larger workshop after the vintage period. The workshop, as I described before the vintage period, is based around simple analyses done well, and the assessments which are available on the Moodle page are straightforward, but they're all based around providing results in an interpretable fashion, which then can be utilised in a winemaking framework or in feedback to the vineyard. Assessment 3 used to be quizzes. However, this year it's slightly different in that we will be tracking a wine. So we'll be following a wine from go to woe, and each aspect of that wine will be discussed for the assessment in wine chemistry terms. So, for example, the grapes, when they arrive, do a sensory assessment of those within a wine chemical framework. And we'll be covering all this throughout the subject. When a sulfur addition is made, you'll describe when the sulfur addition was made, how large it was, the rationale for the size of it. So that's some sort of in the winemaking bounds. But once we've done a pros at wine chemistry, really the lines between wine chemistry and winemaking are blurred, to which they're partner skills. So it's not only, okay, how much sulfur did we add, but what is that sulfur doing within the wine? So what's the chemistry behind that? Then it might be tannin addition. What is that tannin addition doing within the wine actually on a chemical level? If we're tracking a red wine, what polymerization between pigments and tannins are occurring post-ferment? What are occurring during ferment? What bonds are being hydrolyzed by the yeast during the ferment? Pretty much you'll be interrogating the whole winemaking process from a wine chemistry perspective. So it's super interesting. So instead of just thinking about it from a production sense, we're also thinking about, okay, we're producing a wine, but what are we actually doing to the wine such that we have a final product? So what's actually happening below the surface that we can't necessarily see, but will play an important part in the flavour profile and its structure and its longevity down the track. So that's a really interesting assignment. It's you getting super familiar with a particular real-world wine. I really like that assessment. I like it more than quizzes. That's why we're not doing quizzes anymore. However, the quizzes are still available for you to do, and there's a number of them on the Moodle page that you can go through, and they're fantastic revision for the exam. All right. So, any questions? I should say the timing of the workshop will negotiate. So depending on how the vintage is going, so we have a large portion of the class away on vintage. So I'm expecting it'll be shortly after Anzac Day, the 25th of April, will be the workshop. And we'll go for 2 days. And it'll be really good. So we're doing some quite complex analyses, really interesting analyses. We'll be breaking up into small groups. We'll be comparing our results. It'll be really great. Looking forward to it. We've also got the guys from Anton Park coming in, showing their analytical equipment. So these multi-parameter wine analysis engines that they have, where we just have to put in a small amount of wine and it gives us a whole heap of results. Alcohol, malic, TA, pH, glucose, fructose, all the good stuff. Yeah, what subjects are you enrolled in, Michael? Are you enrolled in any other subjects, or just wine chemistry? Okay, what ones have you got? And then we'll get started. You've already done winemaking one. So this class, we may be able to squeeze in an excursion post-vintage. Traditionally, this subject does. We go down the peninsula and have a fantastic visit to a couple of wineries down the Mornington Peninsula. Traditionally, we go to Paradigm Hill, which is run by a former academic, well, engineer, but pretty much an academic. He's absolutely fantastic, and he'll be discussing his wines from, again, a wine chemical perspective. So actually remind me of that, because I'll make sure I get that organised in the latter stages of vintage once all the winemakers start entering the real world again. So I'm going to move on to the subject matter that I'd like to cover. Fantastic. So this is the shortest prac because it's an introductory tutorial and it's an introductory topic. So the concepts covered in that first topic really should be revision for living systems, which we all need, you and me. So what I've decided to do instead is make this interactive. So we're going to go through and we're going to look at redox, reduction and oxidation as it relates to something that we are interested in this case, grape development and winemaking. So we're going to start with a simple case, and then we're going to look at one of my favourite pathways, and I'm going to get you to do a little bit of the hard work for me. So we should be on slide 2 now. So reduction, that should be familiar to redox, reduction and oxidation, where reduction is the gain of an electron. It's the gain of an electron. Reduction, I think for me, being someone who applies science a lot and was never a massive fan of the pure sciences, I did study it a lot, wasn't a massive fan, but I've had a lot more success in applying it. I always like to find an application to a lot of scientific concepts. So reduction, I think, is taught in a first year, in first year of science across the world, very much in the way that things like current and voltage are taught. They're sort of taught as discrete things related to individual molecules or electrons, but not as sort of a broader process. So reduction is often spoken about in how it relates to an individual molecule. So an individual molecule gains an electron. But really you can't have the process of reduction without a whole reaction taking place. So if something's being reduced, then something has to be being oxidized because the electron has to come from somewhere. Oxidation is the loss of an electron from a molecule. But again, I like to think of it as the loss of an electron from a system. So a group of molecules interacting with each other. So let's concentrate on reduction. So reduction is the gain of an electron. the thing that gains the electron is confusingly referred to as the oxidant. So the oxidant gains an electron. When it gains this electron, its oxidation number decreases. We say its oxidation number decreases. And so this is getting into the world of redox potential. Conversely, oxidation acts on a reductant. So a reductant loses an electron. Its oxidation number increases because it actually becomes a more powerful oxidizer after this occurs. Because what do oxidizers do? They steal electrons from the system. So we always talk about reduction and oxidation in terms of electrons. However, and this can be confusing because especially in complex equations, it's hard to see where this electron's coming from, especially when we're talking about redox potential, where most of the time there's not actually more electrons being added to a molecule. It's just that they're taking them from another one which is bound to them. So we'll see that in the case of water. I often think of reduction not only in the gain of electrons, but it is often seen as a loss of oxygen. So when a molecule loses oxygen, more often than not, it's being reduced. If a molecule is gaining a hydrogen, generally that is being reduced. So again, traditionally we always say reduction is the gain of an electron, where a molecule gains an electron, but also often it can be seen, will manifest itself as a loss of oxygen or the gain of a hydrogen. And we'll see that in a couple of examples now where that plays out nicely, and it's a lot easier to see than trying to track where the electrons are going or coming to. Oxidation, conversely, is the loss of electrons or the gain of oxygen or the loss of hydrogen. I really like those three things. I find very easy to remember, especially again when you have complex molecules. It's hard to work out, is there oxidation or reduction taking place. Sometimes I go, where's the oxygen going? Is there oxygen coming in or oxygen going out? Is hydrogen going in or hydrogen going out? And what's fantastic about this subject is in winemaking we often talk about oxidation and we fear oxidation. However, when we start thinking about it in a wine chemical point of view, we start seeing, hang on, oxidation isn't just the formation of aldehydes, which is seen as a taint in a wine. Oxidation is also just the gain of oxygen, the loss of electrons, or the loss of hydrogen from a molecule. So that doesn't necessarily form aldehydes. The oxidation of alcohol produces aldehydes, but many molecules get oxidized, and it's a really important process because it changes that molecule into one, which may be important for our wine properties. I'll be posting up in a couple of weeks' time when we knuckle down into oxidation, a really interesting article about the timing of oxygen exposure to ferments. Fantastic article, and that again is where wine chemistry meets winemaking. So let's have a look. Winemaking begins. Winemaking begins with this equation here. And this is a simplified equation from how it's often portrayed, but a very useful one. Alex, what is that equation showing us there, mate? Michael, just give you a moment to have a think. Water and carbon dioxide combining. That's all right, Michael, you will be good at it soon. And I'm throwing out oxidation and reduction. And but if you haven't yet read the first topic and reminded yourself, it'll all be a little bit confusing. But don't worry, we're going to step through things here tonight. And again, you can use this as a primer for really going through that topic 1. If you can bed down topic 1, you'll be absolutely fine for the rest of the subject. So don't worry. Hey, Sophie, how you going? Welcome. So don't worry so much if you can't, if it's all a little too much at this point, because we'll have plenty of time to get it all together. Trust me. Sophie, there's a yellow circle around an equation there. Are you able to tell me what that is showing us? How you going? Welcome. That's all right. Well, it's a slightly simplified version of something that you will be familiar with, and that's the. I'm just trying to find my. Here we go. You may be more familiar with CO2 plus H2O. Goes to C6H12O6 plus oxygen. Does that sound familiar? Anyone familiar with that one? All you have to do is say yes as well, and it makes me feel better. Oh yeah, we'll get to that. What this equation here is showing is yes, thank you, Alex, is old mate photosynthesis. And it's no problem that you don't remember the equation carbon dioxide plus water goes to glucose plus oxygen. You don't need to remember that because it's only the most famous equation on Earth and the most important one for humankind. So there's no reason to remember that one whatsoever. No, so that's a very important equation. And obviously, being a plant man that I am, I do have a lot of respect for photosynthesis. So what we have photosynthesis is carbon dioxide being combined with water to create carbohydrates with a byproduct of oxygen. So that's what leaves do, chlorophyll, pigment, etc., etc. So the simplified equation for that is above. And why I've got that here is because I think it's a great example of organic molecules, which are covered in detail in the first topic, as well as reduction and oxidation, or redox. And so we'll step through it here. And then we're going to move to a more complicated example of glycolysis. One of the points I want to get out, one of the important points from topic 1, which isn't explicitly stated, but is an undercurrent of it, is that all organic molecules contain carbon. So we've got this backbone of carbon, and they all look pretty similar, except they have slightly different structure. And that slightly different structure gives us subsets of molecules. So we have alcohols, which have like OOH groups on the end. We have esters. We have sort of bases with DNA which have a nitrogen group in them. We have smelly compounds which have sulfur groups in them. What chemistry really is just another language. Molecules written on a page are just like letters in a word. When we add different elements, different combinations of letters together, we get a different word. we have, and a lot of words have very similar structure and they're related, and it's the same as molecules. And once we can start seeing the similarities in different compounds, we can start predicting what they're doing. That is a massive undercurrent of topic 1. And it's important to keep remembering as we go through, because it can be overwhelming when there's just all these different classes of compounds being thrown at us. But what the key to it all is just to sit back and relax and look at the basic structure of the compound and go, okay, this has got a ring structure, so I think it might be aromatic, so I think it might have, this could potentially have color, or it could provide some astringency. But we'll see many examples of these as we go through the subject. For example, in week 7, we'll be looking at all the different alcohols, the manifold alcohols which are found in wine. We concentrate on ethanol, and topic 1 talks about this. When we're talking about wine, we always talk about ethanol, but there's many, many different alcohols in wine. It's just ethanol is the one which has a legal limit to it, and the one which has that classic behavioural byproduct of inebriation that goes with it. So that's the one we concentrate on, but there's equally almost as important alcohols in there which provide structure and different sensory aspects to a wine. Next week we look at sugars, the different sugars, how they get converted into different carbohydrates that we find in fermenting juices. Then we move to acids, then we move to sulfur compounds, then we look at nitrogen compounds. And we finish with phenolics and other macromolecules, and then bringing it back again to winemaking, where we talk about sanitation in a wine chemistry framework as well as oxidation. Should point out as well, we'll be going through the chemistry of sulfur additions as well. All right, so we have this CO2 plus water goes to sugar plus oxygen. And there's a few things going on in there. We have this, let me get a little pointer. Oop. Oh wow, I don't know how to get rid of those. I've now put arrows all over. There we go. We have this equation on the left. 2 waters get converted to oxygen and hydrogen ions. Hydrogen ions are H+. Question is this an oxidation reaction or is it a reduction reaction? So what I'm trying to do here is go back to the very most basic equations, the most important ones in terms of winemaking, because this is how our grapes are formed, how our sugars in our grapes are formed through photosynthesis, but also it's the simplest possible molecules to examine and revise reduction and oxidation. So here we have the classic example of oxidation, the loss of electrons. Whoops, loss of electrons. Remember back to the mnemonic oil rig. Oxidation is loss of electrons. Reduction is gain. OILRIG, oil rig. So oxidation is loss of electrons. So the very first step in photosynthesis, we have water getting split by the energy of the sun into oxygen and hydrogen ions, positively charged hydrogen ions. So why is this oxidation? Why does this count as the loss of electrons? Well, here are the main players we're looking at. We've got oxygen in its natural state on the left, which has 6 electrons and 6 protons balancing it out. So in a normal state, just an oxygen atom would be neutral. And then we have a hydrogen atom to its right, which has 1 electron and 1 proton. And then we'll move on to the next equation, carbon, which has 4. in its outer shell, I should say. So here we have oxygen, 6 electrons, hydrogen 1. When they combine, we get this molecule here. Hydrogen is sharing its electron with one hydrogen here on the left is sharing its oxygen, its electron with oxygen. The other hydrogen on the right is sharing its electron with oxygen, and now oxygen is now in a stable state with the octet structure, which you might remember from living systems, which means 8 electrons in its outer shell. So what we're looking for, what molecules always want to do is balance, and nearly all molecule atoms want to have 8 electrons in its outer shell. And so here, the combination of hydrogen and oxygen sharing their electrons with each other has provided oxygen with these nice, clean 8 electrons in its outer shell and a nice stable molecule that we call water. So this is the molecule that we have on the left-hand side of this equation, water. 2 hydrogen atoms, 1 oxygen atom. They're sharing electrons to bring stability, a nice balanced electron stability. So what happens when this pesky electron comes in with all its photonic energy and splits up water? So this photon comes in and, let's say, for want of a better technical term, blows this water apart. All of a sudden, we have bear with me. We get oxygen produced. Which isn't in yellow. Let me change to black. We get oxygen produced. And in this case, the 2 oxygen atoms are again, in this case, the oxygen atoms are sharing electrons with each other. So now they both have 8. If we go back to the previous slide, we see oxygen in its normal state here has 7 electrons in its outer shell. To get stability, it wants 8 there. So what it does is the 2 of them come together and share an electron with each other, and we get this nice stable O2. Added to that, we get 4 positively charged hydrogen atoms. Now, hydrogen, as we see here, in its natural state has 1 electron when it's neutral. So for it to be positive, it must have lost an electron. So these 4 hydrogens have lost an electron. So there's no electrons sitting here. Over here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Electrons. Our original one here had 1, 2, 3, 4, 5, 6, 7, 8. And there was 2 of those. So there was originally 16. So on this side of the equation, there was originally 16 electrons. When we move to when this light blows the water apart, we now create a molecule with 14 electrons, oxygen, hydrogen atoms which have none, so we're missing 2 electrons. So what this process has done is actually free up 2 electrons But importantly, we have a system which has lost electrons. So that is oxidation. The loss of electrons. So one side of the equation we have 16. That gets converted to 14. So this system has lost electrons. And this is the critical first step in photosynthesis. Those electrons then get carried along what's called the electron transport chain, and they eventually get stored in the form of, well, we'll see, in chemical bond energy in carbohydrate. So does that make sense there, or at least on one level here, that on one side of the equation we've got 16 electrons with 2 waters. Light splits the water apart, oxygen forms and hydrogen ions, and those two only have 14 electrons between them, which leaves us 2 electrons just floating around. That is classic oxidation, the loss of electrons from a molecule. And here it is here, but in a neater way. So on the left-hand side we have water. We've got 2 of those. On the right-hand side we have our oxygen. Here they are sharing their electrons. We have our hydrogen atom, which has no electron, so it's positively charged. And here we've got, well, I've got 4 electrons. That should only be 2 electrons. There, let me erase that. Can I do that? No, I can't. I've attempted to erase it. So that is oxidation. Let's look at the opposite, reduction. So in our original equation, way back here, we had carbon dioxide plus water gives us carbohydrate. The simplified one here is CH2O plus oxygen. So let's follow that through and see if we can count our electrons again. So this time we have carbon dioxide and positively charged hydrogen combining with each other. This is the last step in photosynthesis. This is where we get our sugars produced. So at the start of photosynthesis, we've got the conversion of light energy into electrons in the electron transport chain, and those electrons get stolen from water through the process of oxidation. Now we need to reduce something, bring that energy and store it in the form of carbohydrates, because that's the point of photosynthesis. It takes energy from the sun and it adds it to carbohydrates. So when we gain electrons, it's reduction. So this final step of photosynthesis is reduction. Carbon dioxide is reduced. So this is the Lewis diagram. These are called Lewis diagrams, where you have the element with as signified by the letter, and then dots are electrons in the outer shell of that molecule, of that atom. So we've got 2 oxygens and 1 carbon. Carbon has 4 electrons in its outer shell, so it needs to gain 4 more electrons to get that nice stable 8 electrons in its outer shell. Oxygen has 6 electrons in its outer shell, so it only needs to gain 2. So how does it do this? Well, oxygen shares 2 oxygens, both share 2 electrons with carbon. That's the oxygen on the left and the oxygen on the right. And carbon supplies 2 electrons to the oxygen on the left and 2 oxygens to the oxygen on the right. So carbon's really gaining out through this relationship because it's gaining 4 electrons itself, making it nice and stable. So we've got this carbon dioxide. So in this final reduction step of photosynthesis, we need to convert, somehow get this nice stable carbon dioxide and add more energy to it and turn it into a nice energy-rich sugar. So what do? We get carbon dioxide and hydrogen. We add electrons to the mix, and in doing so, we get carbohydrate and water. So let's follow this through. Let's have a quick look. Let's have a quick look yourself before I drone on and just try and see what's happening with the electrons here. Oops, sorry. Count them out. Count how many there are on both sides. So if you count them out, what we find is there's 16 electrons here, and there's 20 electrons over here. So what we've done is add electrons to this system. So to complete this overall equation, we need to add the 4 hydrogens that are missing. And throw into the mix another 4 electrons. And once you do that, the two sides of that equation balance out. We now have 20 electrons on the left-hand side and 20 on the right-hand side. So what this fantastic last step, reduction step in photosynthesis is doing, is adding energy into this molecule here, which is sugar. So all photosynthesis really is doing is taking electrons and stuffing them into a molecule, which we call a carbohydrate. Carbon, hydrate, water. Carbohydrate, carbon and water. That's what we have. That's what we have here. So again, I know I'm repeating a point, but photosynthesis is converting electrons into energy stored in carbohydrates. And it's this energy stored in carbohydrates which drive everything in grapes from there on in. So if we go back to our original rules, I said we get caught up with oxidation and reduction just talking about electrons. Another way of looking at it is the loss of oxygen. So reduction, is the gain of electrons or the loss of oxygen and the gain of hydrogen. Well, let's have a look at what's happened to CO2 here. CO2 has lost 1 oxygen and it's gained 2 hydrogens. So classic symptoms of reduction in place. On the other hand, oxygen is the gain of oxygen, the loss of electrons, or the loss of hydrogen. And that's what's happened to water here. Water's lost hydrogen, but it's gained an oxygen. So again, they're the things how I remember oxygen and reduction. I try and not just think about it in the sense of electrons, but what's happening to the entire system at play. All right, so they're very simple examples. And now I want to finish with a little exercise just to get you exploring a couple of key organic molecules that we'll be exploring throughout this subject. And it's a really good exercise. It's one that really helped me from taking what I had when I finished uni, which was just rote knowledge, where I just remember things, to actually understanding things. What we have here is the process of glycolysis. Glycolysis is taking that energy that's been stored in carbohydrates, in this case glucose, and using it somewhere. Well, actually, it's two things. I should say glycolysis is two things. One, it's the use of the energy in carbohydrate. And it's also the reuse of the carbon in carbohydrates. So it's two things. So what the plant wants to do, it produces all this glucose, then it wants to use it. And what we're going to see here is every organic molecule that we'll be investigating throughout wine chemistry. This is how they are all formed. Let's have a look at how it works. So we've got glucose at the top here. We've just seen how glucose is formed through photosynthesis. Glucose then gets oxidized to hexose phosphates. At this point, the pathway can either keep going down or it can go to the right. The overall destination of the pathway as it goes down to the TCA cycle, when it gets to the TCA cycle, the job of the TCA cycle is to convert that electron into ATP, which can then be used as actually a fuel source in cells. That'll actually drive reactions. Reactions that require electrons, ATP provides those electrons. They're like the batteries. So one destination for this energy in glucose is to make it all the way down to the TCA cycle where those electrons are harvested and put into the form of ATP, which then drives cellular function. However, not all glucose makes its way there. Some of them get diverted off to create all these other important organic molecules. Look at the first step here. If the pathway goes right, instead of continuing down to make ATP, it goes right and creates cellulose. And what's cellulose, Sophie? What's the role of cellulose? No, well, starch is actually at the top here. So starch is a long-term storage molecule for carbohydrates. So starch is right at the top there. I hear what you're saying, but we don't want to confuse starch and cellulose. Cellulose is structural for plants. Starch is how plants store carbohydrates for then reuse. So starch is stored in the stem of grapevines and in the root zone of grapevines during winter for then breakdown and reuse after dormancy, and it gets broken down into glucose, which then goes through the glycolysis pathway. Cellulose is the structural. It's the structural substance in plants which makes them rigid, provides them support. So leaf tissue is made of cellulose. Stem tissue is made of cellulose. It's the carbohydrate that makes up things like cell walls. So cell walls are composed of cellulose. So that's where some of our carbon goes. Another part of the carbon at this point goes into nucleic acids. So nucleic acids form our DNA. If we keep going down the glycolysis pathway, we get fatty acids, very important. Here we get proteins. Keep going further down through the shikimate pathway, we get phenolics. So here is glycolysis ending up as colour and tannin. Two molecules that we'll be investigating as part of this subject. What is the structure of anthocyanins? How do they form chains? Oh, sorry, what's the structure of tannins? How do tannins form chains to create astringency? How does colour form complexes with tannins, which stabilises the tannin and the colour? We'll be investigating that as well. But that's another byproduct of the glycolysis pathway. Then we go further down and we see such things as citrate, malate, and succinate. Three important, very important acids in wine. Tartrate actually is produced as a breakdown product from a vitamin, and we'll be going through that as well in the acids lecture, but not directly related to the glycolysis pathway. And here we've got another one. The glycolysis pathway goes over to create carotenoids, terpenoids, gibberellin. So what we've got there is flavour compounds, colour compounds, and vitally important plant hormones, all as byproducts of the glycolysis pathway. Every step of this pathway is an oxidation step. Oxidation, gain of oxygen, loss of an electron, or loss of hydrogen. What I want you to do at this point is, topic one is vitally important. So what we're going to do is we're going to stop here, and I'm going to get you to do some homework for me. And everyone who's listening to this recording to do that as well. And I'll post the instructions up in the topic one folder. What I want you to do is select, and what I'll do is I'll get rid of all these. Let's see if I can get rid of these. No, I can't. Let's do those. Here we go. What I want you to do is select a molecule in the main glycolysis pathway. And I'll just point that out once I've finished erasing it. All right. What I want you to do is to select a molecule along here. So we might select pyruvate, phosphoenol pyruvate, triose phosphates, hexose phosphates. And then select a downstream molecule, one which you're interested in grapevine. So for example, Sophie, you might select phenolics. And phenolics all have a basic structure. They have a ring type structure, aromatic ring structure, which you'll see summarized in topic one. So what you do is you would select this and you'd select a grape phenolic compound. And then you're going to answer these questions. What are the molecules involved in your particular arm of the pathway? So that would be what's the first one and what's the last one you're looking at. And what structural change has taken place between this source molecule in the glycolysis pathway and the molecule at the end? So it might be, what is the structure of phosphoenol pyruvate? And what is the actual structure of my phenolic compound? How do they differ? What changes have taken place? So what functional groups have been added. For example, has an amine group, NH4, been added? Has a carboxyl group, COOH, been added? Has it gone from a linear, so a straight molecule, to a circular one? I've got what is the role? What is the role of that molecule? I want you to tell me what is the role of that molecule. Ooh. So what structural changes have taken place? What is the role of those molecules? So what's the role of the phenolics or a particular phenolic you're looking at? What is the importance of the functional groups that are on that molecule? So for example, if it's become circular, what are some of the basic properties of circular molecules compared to linear ones? And you'll find some of those general answers in topic 1. If nitrogen has been added or sulfur has been added, what effect might that have? What's the importance of those groups? Why are they there? And why are these groups important in wine and grape? So why are phenolics important? So you're covering a lot here. You're investigating a lot. You're looking at these critical pathways. You're examining the effects of oxidation reduction. You're looking at functional groups. You're analysing molecules, not just with their name, phenolics, but looking at how many carbons are in there, how are the carbons arranged, what functional groups have been added to those, and why are they important? And then finally, why do we care about them in the first place? If you want to look at malate, what is malate? So we know malate predominantly as malic acid. So what is malic acid? How is it structured and why is it important? This is a really, really important exercise to kick this topic off. From every tutorial from now on in, we'll be looking at set questions, straightforward questions lead towards our exam. But for this one, I want you just as an introduction to explore this pathway and look at organic molecules within the field of wine and grape, so as it relates to winemaking and grape growing. In our next tutorial, which is in 2 weeks' time, you'll need to be up to topic 3. In the next tutorial, we'll be looking at or getting started on our major assessment, the wine tracking. So we'll be strategizing how to get started for that in each of your situations. And if you want to get going early, I have the full guidelines up for that. I have the full guidelines for that assessment up on the Moodle page, as discussed in the first workshop, which is tracking the wine all the way through. But the idea is that each tutorial, we're just going to keep going through, see where we're all at, bounce ideas off each other, and just progress through so we don't have to do it at the last minute and we do a really good job and get a lot out of it. So between now and then, I want you to get really nut out topic 1. Go through that audio and look at those notes. Topic 2 and topic 3. Have a look at the first question set, which is not assessed but are revision questions. Have a look at the readings associated with those and do this exercise. And then we're going to meet up in 2 Tuesdays' time and have a nice long tutorial going through those and going through your assessment. Does anyone have any questions at this point? It's great to get going. I'm really looking forward to doing this subject. I like it because it's very applied. No worries. I think you're going to really enjoy this as we go through. The Pracs after vintage are absolutely fantastic. And anytime you want to come in and do some analyses in the winery, you're more than welcome, beyond that which is required for those first Pracs. Michael, you're coming in tomorrow, I'm pretty sure. Let me know if you can't. Alex, if you can come in tomorrow, let me know. That'd be great. Otherwise, come in another time, and then we'll have Pracs again after vintage. Thanks for coming on tonight. Much appreciated.